Hello, my name is Professor Amanda Bamford. I'm a Professor of Plant Sciences at the University of Manchester in the United Kingdom, and I have interests in air and water pollution. Today I'm going to talk to you about biological monitoring of freshwater systems. My lecture today is going to be all about using organisms to measure the health of rivers and lakes. So the outline of my talk is I'm going to start off with the introduction. We're going to briefly consider biomonitoring and bioindicators and talk about what they are, and also why we actually monitor the organisms that I'm going to show you today. I'm then going to move on to talk about river biomonitoring and how we can use benthic macroinvertebrates to monitor the health of our rivers. And I'll explain all these terms to you later in my lecture. We're then going to move on to lake biomonitoring. We're going to be using algae and zooplankton to monitor the health of lakes, and I'm going to talk to you about how they're useful for doing this. So let's start. Firstly, what are we talking about when we're talking about biomonitoring of fresh waters? Well, biomonitoring is generally defined as a method of observing the impact of external factors on ecosystems and their development over a period of time, or of determining the differences between one location and another. For example, a clean area, a clean lake, and then another lake where you think there might have been a pollution incident. So biomonitoring can be very useful for comparing two different places. So you can biomonitor species or communities by characterising their richness, so that's how many species you find, and the composition, and that means about the type of species or taxa that you find in your system. And this type of biomonitoring has been identified as a very sensitive tool for looking at health of fresh waters. It's very quick and accurate. It can detect changes in aquatic systems very efficiently. So healthy ecosystems can be considered the same as good water resources. It's very important that we have healthy ecosystems in our waters. And the EU Water Framework Directive is based on the principle that healthy ecosystems are the basis for sustainable water resources. So one depends on the other. Since 2014, the National Water Resources Committee in Myanmar have adopted the National Water Framework Directive, the NWFD. And I'm sure that Cecilia has already talked to you about these in the previous podcast. So the EU Water Framework Directive has a number of different elements to it. They measure 5 different parameters. They measure phytoplankton, which are microscopic organisms that contain chlorophyll, aquatic macrophytes, which are plants growing in or near the water. Thirdly, phytobenthos, which are algae living on the bottom of a river. Benthos means bottom. Number 4, benthic macroinvertebrates, which are animals living on the bottom of rivers. And number 5, fish. So all these are excellent indicators for ecological status because they can respond to a variety of different impacts. For example, nutrients, light, temperature, heavy metals, herbicides, turbidity, and water level change. So all these 5 are routinely measured, for example, by the Environment Agency in the UK. So we're talking about bioindicators of water quality. What's the definition of a bioindicator? Well, it's an organism, or a community of organisms indeed, that give information on the quality of the environment. And here we have a picture of a diatom on the left and a macroinvertebrate on the right. So examples of bioindicators. So these bioindicators can be used to biomonitor your system. In a well-studied ecosystem, the changes in the numbers of organisms and types can give very accurate information about pollutants and other stresses from the environment. And across the world, this type of methodology is used routinely. Biological assessment and monitoring of aquatic resources have been measured and used in many parts of the world, for example, Europe, Australia, United States, and indeed my home country of United Kingdom. Many countries or states or water authorities have indeed developed their own biotic indices that reflect the systems in their countries. And one of the references that I've given you at the end of these slides is Li, Zheng and Lui 2010 that give you much more information about all the different assessment and monitoring systems that go on. So why do we measure these organisms? If the number of the species, or in other words the richness and abundance, change over time, it might indicate the effects of human activity on the stream or lake. So you can keep biomonitoring every month or every year and look for changes in your ecosystem. And such biological monitoring is based on the fact that different species react to pollution in different ways. So this is the crucial thing to remember. And the whole of biological monitoring is based on this. Your different species react to pollution in different ways. So why do we survey? Well, one type of survey is surveillance. So you can look at before and after a particular project, a building project, or building a dam, or installation of a sewage system. So you can survey the impact of your activity before and after. The other reason for surveying is for compliance. So by routinely, regularly sampling or toxicity testing, you can ensure compliance with mandatory standards. So you can check that your system, your river or stream or lake, is meeting the legislative requirements for your country. So let's start off by talking about river biomonitoring. So here's some of my students out in the cold in the UK with their waders on, taking different measurements of our rivers. And the ones on the right are doing kick sampling, which is a way of collecting benthic invertebrates from the bottom of rivers. And this is one of the things that you'll be doing or watching doing in our face-to-face activities in Myanmar. So in order to do a biological assessment, it's a 3-step process. Firstly, you need to sample the aquatics organisms, obviously, like these 2 girls on the right, using a kick net. And then you summarise the data using biological indices, and I'm going to show you some of these later. And then finally, you compare to your reference streams or previous data or previous published information to see whether there's been any changes or significant impacts. So this is quite a complicated river food web, so I don't really have to worry too much about it. But what I want to show you on here is that in any freshwater system, you have a pelagic pathway, which is the area where free living, free swimming organisms are found, like fish. And then you have the benthic pathway, which is the organisms that are living on the rocks or the bottom of the river or on the sides of the lake or even on the plants around the lake. So benthic are usually the ones that are attached and not free swimming around. They tend to be found on the bottom of the system you're looking at. So all these organisms in this diagram are potential bioindicators. But obviously there's no point measuring everything, we haven't got the time. And so one of the first things and most common things that people do look at are these types of organisms, which are macroinvertebrates. So now I'm going to talk about using benthic macroinvertebrates as bioindicators. Benthic macroinvertebrates are organisms that inhabit the bottom substrates for at least part of their life cycle, as nymphs very often, and are retained by a 200 micron to 500 micron mesh. So a very particular size of organism. And you have two pictures at the bottom of this slide showing you two common benthic macroinvertebrates. These macroinvertebrates are key components of a food web, and they link organic matter because a lot of them eat the organic matter, and they're actually nutrient resources for other organisms in the food web. These macroinvertebrates are affected by the physical, chemical and biological conditions of the stream to different levels. So different organisms will be affected differently, as I said earlier, which is an important characteristic of a bioindicator. Some are very vulnerable and very sensitive to pollution. For example, this caseless caddisfly shown at the bottom of the slide on the right. While others can live and proliferate in disturbed and really very polluted waters very happily. So taken together, the presence or absence of tolerant and intolerant types provides an insight into the water body's overall health. If we're looking at macroinvertebrates, we have to identify them. And this slide is reminding you or showing you of the taxonomic sequence where animals are classified. We have the kingdom down to the species. So for kingdom, for example, you've got Animalia, and then phylum, for example, Arthropoda, class, Insecta, order, Plecoptera, family, Capinidae, genus, Capnia, and species, Phenalis. Very often, most of us don't have the experience and the expertise to identify something down to species level. Some of these organisms are very, very difficult to identify. Luckily, most times, you'd only have to identify it down to order and family. And in fact, in our lab exercise, we'll mostly only go down to order. So what we'll do is you take your sample, you go out into the river, do your kick sample of benthic invertebrates, bring them back to the lab and identify them down to major taxa, for example, order. And then you look at whether the organisms you found are sensitive or tolerant to pollution. So here's some examples of some pollution sensitive taxa. We have stoneflies, order Plecoptera, the picture at the top, mayfly nymphs, order Ephemeroptera, here with 3 tails, and free-living caddisfly larvae, order Trichoptera, which you have an example of a picture here at the bottom. So if you see in your sample lots of these particular taxa, then you know it gives you an indication that your water quality is quite good. Because if it was polluted, you really wouldn't see these taxa, these orders at all. So it gives you a good indication of the quality of your water. There's some other taxa that are more sensitive. For example, cased caddisfly larva, order Trichoptera, freshwater shrimps, order Amphipoda, and beetle larva, order Coleoptera, which is this example at the bottom right of the slide. So these are called somewhat sensitive taxa. Some of them, like the freshwater shrimps, can actually find in quite polluted waters, but they also are found in clean waters. And then another group will be your pollution tolerant taxa. Now these, if you find a lot of these taxa, you know that your water is probably polluted. Obviously we don't know by what at this stage, but just the presence of these organisms are a good indication that your water quality is not very good. So you have things like midge larva, order Diptera, family Chromidae, these ones here, black fly larva, order Diptera, and leeches, order Hirudinea. So if you see a lot of leeches in your water, you know the water quality is very poor. So hopefully by now you're getting the idea of how you can use organisms as bioindicators. So not just by looking at the species or the taxa that are there, you can actually do some calculations of metrics to help you analyse your system. And there's lots and lots and lots of these. Some are more popular in, for example, the US, some are more popular in Europe, and some are more popular in Asia. I've listed here some of the most common ones. We have the Biological Monitoring Working Party Score, in other words the BMWP, and this was developed in the UK and is used extensively in the UK and in fact around the world. And this system allocates different taxa a scoring according to how polluted the organism can stand the system. So for example, something very sensitive to pollution would get a score of 10, and something that's very tolerant to pollution might get a score of 1. And we're actually going to look at this BMWP and try it for ourselves in the lab. The other metrics you can look at is total number of taxa, so the total number of species or genus or order that you find, so that's richness. The other thing you could do is look at the number of Ephemeroptera, Plecoptera and Trichoptera taxa that are present in your sample, and these are the ones that are very sensitive to pollution. So if you have a lot of these, then you know that you've got quite good quality water. The other one you can look at is the percent of the mayfly, which is the Ephemeroptera taxa, and look at that as a total number of your taxa, what percentage are mayflies. So there's a number of different metrics you can use. So for example, some of the changes that invertebrates show when there's been an impact. Nutrient enrichment. If you have nutrient enrichment, very often you see an increase in the ratio of aquatic worms to aquatic insects. So that might be something very straightforward that you could look at. Another metric you could look at was increased ratio of midges, in other words chironomids, to other aquatic insects. Low low dissolved oxygen, if you have that in a system with very low oxygen levels, you might see an increased ratio of aquatic worms to aquatic insects. Contamination by heavy metals, you might see an increased ratio of midges to other aquatic insects. And finally, low pH, you might see a loss of snails, daphnids, mayfly, midges in a system with low pH. And in fact, in our workshop, we're going to be looking at pH and we're going to be looking at nutrient enrichment to see what impact that has on the benthic invertebrates. What are the advantages of macroinvertebrates? Well, there's a lot of advantages of using macroinvertebrates to biomonitor your system. They're found worldwide. I'm sure I'll see invertebrates that I see in the UK. I'm sure we'll see some of them in Myanmar. So they're found in habitats all around the world, and they're very, very similar all around the world. They're found in areas that might not have fish. So again, that will be very useful, because if you don't have any fish in your system, there's no point using fish as your bioindicator. There's a large number of groups. So as I said in the previous slide, you can look for a wide spectrum of responses, and they can tell you a lot about what's happening in the system. They're sedentary, they're not swimming around, they tend to stay in one part of your river system, so you can keep going back to the same area again and again, maybe throughout the year or over years, and look for changes. Their life cycle is long enough to see changes, but quick enough to detect problems fast. The advantage of using bioindicators like this is that if you try to use chemistry methods to detect a pollution incident, for example, that pollution incident might have happened a few weeks ago or a month ago, and now the river quality in terms of that particular, say, heavy metal is now gone. The heavy metal has disappeared, but it had an incident a few weeks ago that was quite detrimental. If you just use chemistry methods, you might miss that. But the macroinvertebrates have been sitting there and they've experienced, for example, that heavy metal impact. And you'll be able to come along a few weeks later and see that impact. Maybe some of the species have now disappeared, maybe a lot of them are dead. So you can pick up changes that might have happened a while ago. But also they react very quickly. So say an incident happened the day before, you can go the following day and you probably see impacts already happening on the organisms that you find in that system. The sampling analysis is relatively inexpensive. You don't need lots of very complicated equipment. And the taxonomy of the many groups are well known. There's lots of keys out there to help you identify. Some online, there's some really good ones online. There's lots of books to help you identify the different groups. There's lots of methods, data analysis. There's lots of literature around using this method of biomonitoring. And the response of very common species is well known, so you could look up the species that you're finding and find out their responses to different pollution impacts. However, there's some disadvantages. Under natural conditions, currents and substrates, some of those might still affect the distribution and abundance. It might be a natural change that's happened, not a human modified change. There's seasonal variations, diversity anyway, natural variations in life cycles and food webs. And so these can create problems if you're not aware of them. And then obviously pollution might not always be the one responsible for the absence. Maybe it's just not found that time of year. Maybe something's happened to the habitat that's nothing to do with pollution at all. Sample processing and identification can be costly if you have to get somebody else to do it or pay someone else to do it, get an expert in. And it can be difficult identifying the species, as I'm sure we'll find out, and can be very time consuming as well, especially if you have lots and lots of organisms in your sample. So this is a case study published in 2007, looking at the shifts in community composition related to human water withdrawals along a river in the Oregon, United States. So this is to give you an example of how this type of biomonitoring of benthic organisms can work. On this graph, we have relative abundance on the left hand. We have water withdrawal going along the bottom axis, and the line is the amount of water that's discharged from the river. So first of all, you can see the amount of water extraction from this river means that by the end of the system, end of the time, there's very little water left in the river. So the water withdrawal will have a major impact on the community living in that river. So the green bar, the bars along here, the green bars represent the disturbance intolerant insect taxa that we were talking about before. For example, the Ephemeroptera that are very sensitive to any disturbance, to any pollutants. And you can see here that as the water is extracted, the relative abundance of these disturbance intolerant, these pollution sensitive species drops dramatically. However, in contrast, if you look at the blue columns, which represent the adapted non-insect organisms, for example, leeches, these increase as the water levels drop in the river. So you can look at community changes very, very quickly by monitoring the composition of the species found in your river. So let's move on now to lake biomonitoring. Here we have pictures of some diatoms, and we have one of my students with a net, with a phytoplankton net, which is the net you use very often to collect algae in lakes. So there's a lot of pressures on freshwater lakes, as Cecilia discussed in her earlier podcast. These main pressures include excess nutrients and organic pollutions that can lead to eutrophication, as you can see here in this picture on the right, and pesticides and herbicides runoff, all that can impact on our lake systems. So instead of using macro invertebrates, in lakes we can use algae, phytoplankton, as our bioindicator. These are microscopic organisms, and they contain chlorophyll and require sunlight in order to grow and live. And they're primary producers in our lake food web. And here we have some examples of some algae that you find commonly in lakes. Phytoplankton have very short life cycles and rapid reproduction. So that means that they react very sensitively against any changes, especially nitrogen and phosphorus. And one thing to look out for when you're doing your lake sampling is cyanobacterial blooms. There are naturally occurring cyanobacteria or blue-green algae, and they can quickly multiply and form dense mats, just covering the whole surface of your lake. And they most commonly occur at high temperatures and with high nutrients in the water. And worryingly, some of these cyanobacteria produce cyanotoxins, and each genera can produce multiple types of cyanotoxins. So, if you're doing a lake monitoring system, obviously cyanobacteria are one of the key organisms that you're going to look out for. At the bottom of this slide, you can see a picture of Lake Erie, and you can see all this bluey-greeny colour, which is the Microcystis bloom, which is a very common cyanobacteria. The bloom is like a greenish, thick, paint-like material here. You can see here on the slide at the bottom. Another common cyanobacterial bloom is caused by Anabina. And this bloom tends to occur in the summer, and it's slimy, and you can see it as large dark dots in the water samples when the levels get really, really high. A third type of cyanobacterial bloom is caused by Plankthrix or Oscillatoria, and this is filamentous bacteria that cause horrible, strong, earthy colour, earthy smell in the lake with dense surface scums on the top. So when the levels of these cyanobacteria get really high, you get these blooms. And this is what they look like under the microscope. Here we have Anabina on the top left, Microcystis, and Plankthrix or Oscillatoria. So we'll be looking out for these when we do our lake analysis, because even if you have just a few of these phytoplankton in your sample and it's not yet a bloom, just the very presence of them will give you cause to concern, because you don't know that maybe later on in the year, or when there's a sudden influx of nutrients from a fertilizer, that these cyanobacteria suddenly multiply rapidly, produce cyanotoxins, and give you a real problem in your lake. They can cause lots of human symptoms: indigestion, vomiting, diarrhea, abdominal pain, headaches. So these different algal toxins, there's a whole range of them here, as you can see in the table on the left, from microcystins produced by Microcystis, on nodularium produced by Nodularia. And some of them can produce more than one algal toxin. So these are not things that you want to build up in your lake. The World Health Organization has a recreational water guidance and action level for cyanobacteria and cyanotoxins. So these are things that are very routinely monitored worldwide. So you have, at low levels of these cells, you have low probability of acute health. But if you count numbers of your cells and they're above 10 million per mil, which they can reach, the probability of actually acute health effects is very high. So what you routinely do in your monitoring is to count the number of cyanobacterial cells in your sample. And if you count your number of cells, you can estimate the levels of your cyanotoxin in the sample. There's also drinking water levels, and again these are routinely monitored. The amount of microcystins in the water and drinking water needs to be monitored, especially in your areas where you've got algal blooms. You don't want your cyanobacteria levels to be very high. So that's phytoplankton. So we've now covered macroinvertebrates in your rivers. In your lakes, you can use phytoplankton, and you need to look out for cyanobacteria. You can also look at zooplankton as your lake bioindicator for health. Now these are minute aquatic animals that are non-motile or very weak swimmers. They can move around a bit and they drift in the water. That's what plankton means. It means drifters. They're an important element of the food chain because a lot of fish eat these zooplankton. They're a form of fish prey. And the zooplankton community is composed of highly sensitive organisms that respond to a large number of environmental changes in a relatively short period of time. It can even be hours. So let's have a look at the major zooplankton that we could actually monitor. Firstly, there's cladocerans, or we call them in England water fleas. You usually find them any standing water, ponds, lakes, anywhere like that. And they're important food for fish. They feed on smaller zooplankton, some species feed on algae, and they're very highly responsive to pollutants. Second group is called rotifers, and these are soft-bodied invertebrates with a very short life cycle, and they float around in your water body. And under favourable conditions, you can get very high quantities of these very quickly, so they're very responsive. Finally, there's copepods. These organisms with a tough outer skeleton, and they can swim really fast through the water. And they can be very tolerant to a wide range of pollutants. And there's two major groups of these. There's cyclopoids and calanoids. And you're very easy to tell apart by the size of their antennae. And because they're tolerant to a wide range of pollutants, looking for these in your lake sample can tell you a lot about your water quality. So sort of metrics you can use for zooplankton, if you see a lot of zooplankton, this has been related to rising levels of eutrophication, lots of nitrates and phosphates. And the ratio between the two different types of copepods has also been associated with disturbance levels, often nutrients into rivers as well. So you can look at the ratio between these two different organisms to give you a number to assess your lake compared to another lake, for example. So we're now seeing three different bioindicators classes: macroinvertebrates, phytoplankton and zooplankton. So how do you choose which one do you do? Do you do all of them? Do you have enough time? Well, the ideal indicator should be easy to measure and interpret, be sensitive to before any severe damage to the whole system takes place. So you want to pick up any major changes with these bioindicators before the whole system is affected and maybe all your fish, for example, start dying, or it affects human populations. You want your bioindicator to be sensitive to a range of stresses and levels, not just one pollutant, applicable across water types, different types of systems. Ideally distinguish between natural variation and human disturbance, and contribute to the diagnosis of the impact or change. So it give you a diagnostic level along with all your nutrient, chemistry, pH, oxygen levels. This is an added indicator for your water quality and your ecosystem quality. I've given you here some very important resources and references that you can go and look up, especially the one at the bottom, which really talks about water quality criteria for lakes and rivers in Myanmar. It's a preliminary report. It's only been published in 2017, but it's really useful reading because it will go through a lot of the things that I've been talking about. So to summarise, you need to choose your bioindicator and choose your system that you want to, according to what system you're looking at. So in our workshop, hopefully we'll have time to look at all these types of bioindicators, and you can have a go yourself and see how it works. I look forward to seeing you then. Thank you very much for listening.